Good afternoon, everyone. I hope you had a good lunch. And for the second half, I'm going to guide you through the web interfaces of ISMARA and CREMA, how to upload the data, what kind of data you can upload, how to do it properly. And also in the end, I will show you how to use averaging feature of ISMARA in order to calculate different interesting things, how to average replicates, how to calculate contrasts between different sample subsets. All right. So first I would like to start with a table which shows what organisms are currently supported by ISMARA. So in addition, in this table, you can see different statistics on how many motifs, how many transcription factors, what genome versions are supported. And here I must note that we show here on the latest genomes supported by ISMARA. For example, for human and mouse, there is also support for MM9 and MM10 versions of the mouse genome. The same for human. We also support AG18 and AG19. And now about the supported data types. So ISMARA is designed to analyze gene expression data, and either it accepts results of next generation sequencing, either in form of raw reads in FASTQ format or mapped reads in BAM or BET format. ISMARA also can process microarray data. So it was originally designed to support Affymetrix microarray chips. And we have now support for various chips for human, mouse, rat, yeast, and E. coli. But potentially, if you would like to have support for some chip for our organisms, which is not yet supported, please contact us so we can work on this. So first about how we process microarray data if we obtain it. So first of all, we make correction of the background and also remove unspecific binding using different bioconductor packages like AFI, oligo, and GCRMA. Then we filter out probes which are not expressed, quantile normalize the expression values, and finally we log transform the data, and in this form it goes into ISMARA. In case of raw read processing, for RNA-seq, we first map reads to the transcriptome with Callisto program. When we count how many reads we have per transcript, which was shown by Eric in the morning, we calculate TPM values for every promoter, log transform the data, and when we analyze it. The same for ChIP-seq, with a difference that, first of all, ChIP-seq data we map not to the transcriptome, but we map it to promoter regions. And also, instead of calculating TPM values, we just quantile normalize the counts of reads which fall into the promoter regions. But since release of the CREMA, ChIP-seq support in ISMARA is absolute. So if you want to analyze your ChIP-seq data, so we strongly recommend to use CREMA and not ISMARA. So also, it's possible to upload not the raw reads, but mapped reads, like in BAM or BET format. So, and in this case, we just use absolute genomic coordinates to map, basically to count reads in the transcripts. Then again, we calculate TPM values for every promoter, log transform the data, and process it. We strongly recommend, if you have a choice, to submit the raw data instead of map data, just to keep a consistent processing of the data within ISMARA pipeline. So what kind of file formats are supported. So these are cell, BAM, BET, and FASTQ. And it's very important that files have the correct file extension, because in ISMARA, files are recognized by extension they have. In addition to these extensions, ISMARA also supports files which are compressed in different forms, so like zipped, tart, zipped, and so on. Actually, you can submit archive which contains, for example, FASTQ data or BET data on cell files, and when such archive will be automatically decompressed, files are going to be extracted and properly analyzed. What is also important when you submit map treats, like in BET or BAM format, please make sure that this data was mapped to the genome, to the same genome version which is used by ISMARA. Because if your data were mapped to, let's say, AG19 and you run ISMARA with AG38, of course it's not going to work properly. Also, does it make sense to compress everything as much as possible? So certainly it makes sense for files like cell, BET, and FASTQ, because compressing these files, you can significantly reduce the file size, and reduced file size, of course, minimize the upload time and probability of network errors, which can happen during upload. It makes no sense to compress BAM files, because BAM files are already compressed. And usually there is no benefits to compressing all files into one archive. So if you have just a bunch of FASTQ zipped files, so please don't put it into one big zip file, so it doesn't help. All right. Okay, now we are coming to a very important point about good representation of your data in the on the report pages. So underneath, so at the bottom of the slide, I have two examples. So this is essentially the same activity profile, which is shown for samples sorted in different order. And I'm sorry, I have missed one important point. So when you upload data to ISMARA, ISMARA takes the file names as a sample names. And all the data later on, for example, on motif activity profiles, are basically represented following the alphabetical order of these file names. And in case on the left, you can see basically a profile which has proper file naming, proper sample naming, and they are sorted correctly. And you can see there is a clear difference between, for example, day 0 and day 3. And if you had the same files named differently, so the order is broken here. And in this case, you have profile, which is very hard to interpret unless you take an effort to really trace back what exactly are property of each sample in the data set. So basically, we are following rules how to name the FASTQ files. So the names should be relatively short. They should have intuitive meaning, and also, desired order of the samples which you would like to see in the report should follow the alphabetical order. Also, one important thing about very long sample names is that when they are shown, for example, on the plots, very long names get truncated, and which also reduce the ability to understand data correctly. Okay, and here's some example of good naming scheme for the samples which you upload to the web server, and bad example. So in one case, we have some comprehensive, relatively short names, which are self-explanatory. And on the right, we have just file names with some identifiers which are unclear. This is what usually you can obtain from a database, but it's really hard to interpret later on. So I strongly suggest that you take some effort before uploading the data and rename the files correctly. So for people who use Linux-based machines, the easiest thing is just to create symbolic links to the files with desired names, and then use these symbolic links to upload the data. All right. And the last thing, also there is a trick to enforce the desired sample order by using numerical prefixes in the sample names. And on the left, I show some kind of good example, so where you have numerical prefixes and they just follow the desired order. And please pay attention that there is a leading zero in the file name. And this is very important because without leading zeros, the order will be broken again. And for example, the sample with which starts with 14 will come ahead of sample which starts with 1. So using such tricks, you can easily obtain very nice representation of your results, which will help you to explore and extract some useful information from the analysis. Next part about file naming is about requirements for proper paired and FASTQ file processing. So paired and FASTQ files require a special suffix at the end. So it should be for first read in the pair R1 suffix at the end, and for the second read, it should be _R2. And ISMARA understands only these suffixes. So if you have paired end data, please make sure that your FASTQ files have the correct suffixes at the end for proper processing. All right, so let's say you have prepared all your files and you would like to upload it to ISMARA server. So you go to web page, and so here is actually a submission interface. First of all, you can provide your email address. It's not required, but it's strongly recommended, because in case of any problems, so for example, your data set fails for some reason or upload was not complete, so in such cases, we can contact you and actually help to resolve all the issues. Second field is a project name. It's also optional, but again, it's strongly recommended, especially for people who run multiple analyses on ISMARA. And project name will later on incredibly help you to distinguish between results pages, because on result page, you can see the project name on the top. And so you're not going to mistake one analysis with another one. So the next row is a selector for data type. So you can choose microarray, RNA-seq, and ChIP-seq. And then there is also selector you can choose if you would like to run your data set with microRNAs included or without microRNAs included. So in order to upload files, you just need to click Upload Files button. It will bring you a very simple interface like, shown here, add file, start, cancel. And also in case if you if you like to upload next generation sequencing data, it will show you a selection where you can choose which organism version you would like to use for the analysis. So, as I said, so we have here selection, human, mouse, zebrafish, and so on. So how to add data for upload? So you just click this Add Files button, so you get file selection dialog, and in this file selection dialog, you can choose multiple files at once, add them to the interface, and once they're added, you just click Start Upload button, and yeah, everything starts to upload. Actually, in this window, you can see a progress, so which tells you approximately how much time the upload will take. With ISMARA, you can upload also links to the data files instead of real files. So in order to do this, you just need to select the second button, which says Upload File Links, and there you will have a text input field in which you can paste a list of links which point to the data files. So the rule is it should be one link per line. And once you have all your links in this input field, you just click Submit Links button and everything sent to the server. What happens next? Actually, on the server, all your files will be downloaded. And once download is complete, yeah, the analysis will start. And the third option is to, instead of links to the files, you can use database IDs from Sequence Read Archive database. So they are shown here. So they start with SRR prefix. And again, you just need to paste these database IDs, one per line in text input field, and then submit it. But here we have an extra feature which can make your life much easier. So for every database ID, you can basically add also a little, a desired sample name. And in this case, downloaded files will be automatically renamed just to give you a nice representation of results later on. All right. What is important about submission of database IDs is probably worth to mention here that sometimes connection to database is relatively slow. And for data set which contains large amount of ASRR IDs, download can take really long time, much longer than analysis itself. So in this case, please just be patient. Okay, so I have shown already how to upload the data for the web interface. So it's a very simple solution. You just go to the page, add files, click button, everything starts to load. But the problem is that it requires that you should have your files on your local computer. Sometimes data sets, especially next generation sequencing, will be extremely large, and it's hard actually to first transfer and even store them on the local machine. In this case, there is a second option. This is ISMARA uploader script. So this is a script which can be run in command line through the, for example, SSH session directly on data storage machine. And this is a very good alternative to the web interface. It's usually more robust, less vulnerable for network errors while uploading large data sets. The only thing is that it's a little bit more complicated because it requires some knowledge of Linux command line, and it also requires some Python environment available on the machine.
Mikhail,
Yes.
there is a question about if you're using SRR with paired end reads, how should you do this?
Yeah. Good question. So the thing is that so database ID, it basically corresponds to some database record, and in case of a single end, it will just contain single FASTQ files. In case of paired entries, it will contain basically both FASTQ files for the first end and for the second. So basically, our tool, in case of paired end sequencing, will download two corresponding FASTQ files if corresponding database record is for paired end data. So users shouldn't worry about this. So they just give database ID and that's all. And then already on our side, we properly decide, yeah, if it's paired end or it's single end. All right. So a bit more about Uploader, which could be also useful for people who would like to integrate it into some pipelines which we have, because it can be embedded into some pipeline processing for automatic data submission, and later on with some, you can also add some parts for automatic data retrieval. So it's a simple Python script. It provides all the functionality of web interface. So basically, you can upload exactly the same things, exactly with the same options. And software environment which required for this script is very simple and can be easily installed with, for example, Conda package manager or with other package managers which you like. And yeah, I try to basically show some standard usage scenario when, for example, there is a user computer, you connect to the remote data server where your data is actually stored, and you run this script in order to upload data to ISMARA server, and then you can go to the ISMARA server and see the results of the analysis. So basically, what kind of input required for this script? This script, first of all, requires a file which contains information about files you would like to upload. So it should contain paths to the files. In this case, in example, I show paths to FASTQ files. So again, it should be a single path per line. The same rules about naming applies as before as for web interface. So you just have a file with file paths, and that's enough. But in addition to file paths, you can actually also supply a list of links, if you like, or list of database IDs. So exactly like in web interface, you can provide different database IDs and even annotate them with desired sample names. So, and here basically example how to run the uploader script and what kind of options it accepts. So basically you use the following command, know-how Python ISMARA uploader. You can specify your email. Again, this is optional. You can skip it. You specify your project. You need to specify the data type, RNA-seq, ChIP-seq, microarray, and then you can specify the organism ID or genome version. So for example, it could be human, mouse, or you can specify that it should be MM39 or MM10 if you like. You can also specify if you would like to have mirrors included of the analysis. And finally, you provide the file list. So when you start the script, this script starts to upload the data to the server. And once upload is finished, it actually will return a link to the ISMARA server page, which should contain your results. And if you execute command like this, these links will be saved in results link file. And so you can check GitHub page for documentation, examples, and download script and start to use it. So once data is uploaded to the server, either through web interface or through the uploader script, the first thing which appears is the ISMARA status page of your project. So very important thing here is that people who do not provide email address when they submit a job, they should save this URL somewhere in order to check your data later on. Because people who submit data with email address, they will get an automatic notification once analysis is finished. If no email address provided, the only way to see the status of the job and see results when analysis is finished is to check the URL of the status page. So once the processing is finished, actually this page will be automatically updated and you will see the ISMARA results page instead of the status messages. Also, in case of any errors during the computational analysis, so these errors will be shown also on the status page, and in case if you see something, you can immediately contact us and ask for help. All right. Okay, this I have already described. Now, how long does it take actually to run ISMARA? So I would say that normally, on average, ISMARA runs from 1 to a few hours. Of course, sometimes people submit very large data sets. In this case, running time can be up to 1 day, but normally it's 1 to 4 hours. And in case if analysis is not finished in 24 hours, most probably something went wrong. And in this case, we suggest that you contact us. But also, I must say that we regularly check content, we check our server for errors, and if we detect any errors, we also try to contact users in such cases, in order to resolve the issues. Okay, it was a question already about confidential data, so a few things about how actually data stored on the server. So first of all, results now are kept on the server for 6 months. So this is a guaranteed time. After 6 times, we may delete the results folder just to free the disk space. So the user input data is actually removed as soon as analysis is complete. So we don't keep user data on the submitted user data on the server. Every ISMARA run is saved under unique URL, and basically only person who knows the URL can access it. And as Eric mentioned, so we have also ISMARA version with extended security options, which require license and which include password protected access, isolated storage, restricted access to the data. So potentially, we can also work with confidential data.
Mikhail?
Yes.
There's a question in the chat. How can we cancel our own job?
Okay. In fact, you couldn't. Yes. So jobs right now it's not possible. But again, if you think that something very wrong happened, yeah, you can always just mail to me, and yeah, I can check what is going on the server, and yeah, if there is something wrong, I will fix it. All right. Now we move to the overview of downloads which are provided by ISMARA. So what kind of data you can get in flat file format for follow-up analysis on your own. So first of all, downloads are available on the left of the main results page. So you can see I have highlighted it this in the red, and there is a list of files which you can download. So these, first of all, activities, activity error bars. You can download regulatory interactions inferred by ISMARA, significance table. You can also download the expression table, which was used for ISMARA analysis. And in the end, you can download the whole report as a compressed archive. So first of all, about activity table, so we provide data in the form of TSV file. So this is just simple text file, ASCII text, with all values tab separated. And yeah, the format is very simple. So as a header line, we have motifs. As first column, these are the sample names. And basically every value corresponds to the motif and sample pair and represents the activity which predicts expression change in corresponding sample. Yeah, if you have one binding motif of a corresponding one binding site of the corresponding motif. The same applies for the activity error bars table. So activity deltas table, so it's has exactly the same structure, but instead of activities, it just contains corresponding error bars, again for every motif in M in sample S. And so, for example, these tables are used to build to draw the activity profiles, which Eric has demonstrated earlier. So you can do it also yourself and yeah, play, for example, with order of samples or make some changes and so on. All right, regulatory interaction. So these are the tables which actually contain information about interaction between motif and its target. So this is a big tar archive which contains one file for each motif. So they are named correspondingly inside the tar archive, and inside each file you have a table which contains 4 columns. The first column contains a promoter name. Then it contains log likelihood score. Then motif name, which interacts with given promoter. And finally, it also contains information about transcripts which were associated with given, so which are actually driven by a given promoter. So in this case, for example, we have a promoter which is located on chromosome 19, and it drives the single transcript, yeah, from cytochrome P450 gene. Then we also provide a table which contains information about motif significances. This is basically the table which you see on the main results page. So, and it's very simple. It has only 2 columns. So you have a column which contain motif names and column which contains significances. And And yeah, one thing is that this table is already pre-sorted. So on the top, you see the most significant motifs. And yeah, just to remind you that motif significance is essentially a squared Z values, average and square rooted. All right. And finally, not finally, we also provide expression tables, which contain promoter expression values. So again, this is a tab separated file, which we have expression values for every promoter sample pair. So for rows, you have promoters, for columns, you have samples, and values are logged to TPM values. So in case if you would like to also do some analysis on expression, you can use this table. And finally, you can download also the full report. And this is a tar archive, which contains basically all report HTML pages, all pictures, these downloadable files. And on these web pages, you basically have almost the same functionality. So you have interactive pictures, you have tables which you can sort and in which you can search for different terms. The only things which are missing in local copy of ISMARA report are, so these are the gene search function, which is not available because it requires a server. Then page with promoters sorted by fraction of explained variance is also not available. And it's also not possible to use local copy of the report to perform sample averaging. All right. So I think that was about ISMARA. So are there any questions? No, okay. In this case, let's move now to CREMA. So like for ISMARA, here I show the list of species which are currently supported in CREMA. So it's only 4, and we have no variety for genome versions here. But I had this question already previously in the chat, so I can say that updated version of human and mouse HG38 and MM39 will be added to CREMA very soon. Yeah, and basically, yeah, they use exactly the same collection of motif and transcription factors which we have in ISMARA. All right, so what kind of data is supported by CREMA? So currently, CREMA supports only next generation sequencing data in FASTQ format. And also supported data types include either chromatin accessibility data from ATAC-seq or DNA-seq experiments, or ChIP-seq histone modification data like different epigenetic marks, methylation or acetylation marks, and so on. So what is important, unlike ISMARA, CREMA requires one extra additional file for every data set to be submitted to the server. And so I'm going to explain what is it right now. So for every data set, we expect some sample annotation file, which we so we expect that it's going to be called samples TSV, and it contains information about associate basically files with some condition names, and moreover, it should also provide information what kind of data is in this file. So I'm talking about this second column, and this is especially important for ChIP-seq experiment, where we should know which files are coming from correspond to foreground in ChIP-seq experiment and which files correspond to the background in the ChIP-seq experiment. Otherwise, this is very flexible way to submit the data in the sense that, first of all, you don't now need to rename files anyhow. You shouldn't care about adding any suffixes for paired end or single end data. So everything is defined by the names which you provide for the samples in the first column. Moreover, it is also possible to mix now, for example, single end and paired end data. It's possible to have multiple FASTQ files per one condition. So in such case, for example, here I have two FASTQ files for condition 1. So these files basically are going to be merged. So also the last column for FASTQ file 2 is optional. So in case if you have single end data, you just ignore the last column and leave it empty, and that's all. So basically for every data set, you need to provide a file with description in order to CREMA to perform the analysis. All right. So I basically said this already. Yeah, multiple files per sample and mixture of paired end and single end data. All right. So, and about naming rules, as I already said, it's absolutely not required that you renamed your files anyhow, so they could have any names. But for sample naming, we ask user to use the same rules as for CREMA. So sample names should be comprehensive, they should be relatively short, as short as possible. And also in case of CREMA, order of sample names in the plot is defined by order of sample names in samples TSV file. So you can very easily define the desired order by just jungling with rows in this file. All right, now we go to the CREMA web interface. This is quite similar to what we have for ISMARA. So again, we have email and project optional fields, which we strongly recommend to fill for exactly the same reasons. So in case if something goes wrong, we can contact you, we can try to resolve a problem. So next step, you have to select what kind of data you have. Either this is accessibility data or this is a ChIP-seq data. Then you have to specify the genome version, which is going to be used for the analysis. And once it's finished, all you need to do is just to click this Add Files button. And then you need just to add the files, like in ISMARA, but yeah, please don't forget this samples TSV file, which will be present. So basically, you upload your samples TSV file, you upload corresponding FASTQ files, and essentially that's all. Like in ISMARA, you also have a progress bar, which will tell you how much time your upload is going to take. So it's quite similar. And along with uploading real files, CREMA also accepts database IDs and links, and it all done using these samples TSV file. So instead of file paths in this file, you can actually give, for example, SRA ID, like it's shown here. Again, in case, you shouldn't worry about this. If this is a single end data, it will be automatically recognized. If it's paired end data, it's also going to be automatically recognized. And also the same way you can add links instead of file paths, and like ISMARA, CREMA is going automatically to download files either from database or from internet, and once upload is finished, it will run the analysis. Moreover, with this file format, it's actually possible even to mix different types of data. So you can upload, for example, only 2 FASTQ files, and the rest you would like to obtain from database. So yeah, it's all going to it's all going to be done automatically on CREMA server to downloading things from first database and then from the internet links. All right. Yes. So we also provide CREMA uploader script, which can be used from the command line. And as before, I can strongly recommend it for people who work on large data sets, which who keep the data on remote data storage system. So you just use the script to run it from command line on the remote machine, exactly the same way as was shown before for ISMARA. And basically, it has exactly the same requirements for software to be pre-installed. So it's essentially, you just need a Python and couple libraries, and that's all. And of course, all details you can find on GitHub page. And here again, a little demonstration how to submit your things. So you again, like in web interface, you can supply your email and project. You need just to select what kind of data you have. Either this is epigenetic marks, or this is chromatin accessibility data. You select the genome version which to use. Then you provide this samples TSV file as input, and after upload is finished, your link to the results, to the status page, at first is kept inside these results in file. Yeah. Sorry, this I already explained. Okay, so CREMA status page is has exactly the same function as ISMARA status page. So while your job is running on the server, it just shows the status, either computing, or for example, if first it needs to download something from internet, it will show you which files it's downloading currently. In case of an error, it will just show you some error. Yeah, and again, in such cases, you can contact us to ask for assistance. Okay. Now about running time. Actually, I made a mistake. Okay, so CREMA running time is actually much longer than ISMARA, because while for ISMARA we use very fast pseudo-alignment algorithm, in CREMA we have to read to map reads genome-wide, and sometimes it takes quite a long time. And on average, I would say that small data sets require at least few hours, but for large data set, it will be a few days in order to run the analysis. So in general, if your data set has a reasonable size, I would say around 20 samples, and it's not finished in 48 hours, then yeah, it might indicate that something went wrong, and you can contact us. Yeah, so we will check the status of the job. All right, so now about downloads. Like before, all downloads are listed on the left of the main page, of the main results page. And yeah, here what we have. So first of all, we provide users with CRE, list of regulatory elements. Then we also provide table which contains signal intensities of series across samples, activity and activity error bar tables, regulatory interactions between motifs and regulatory elements, table of motif significances, and finally you can download, like for ISMARA, the whole report as a compressed archive. So CRA list, which we provide, so this is again a text file with tab separated values, which contains the following information. First, these are coordinates of a regulatory element. It also contains length of the CRA, then CRA ID, and then it also has information about closest TSS. So in this case, this is this transcript, which is closest to the regulatory element. And then for this transcript, there is additional field which contains information about transcript, including distance to the regulatory to this regulatory element, and also association probability, which was calculated, like Eric has shown in the previous session. When signal intensity table is very simple, these are just log of normalized read counts, which are given per regulatory element and per sample. Activity table, exactly the same as for ISMARA. So for every motif and sample pair, we actually have activity value. The same about error bars table. Again, so this is simple text file with tab separated values and with error bar, which correspond to sample and motif pairs. And regulatory interactions file contain again, so this is a TAR archive, which contains a set of files, one file for each motif. And inside this file, we have, again, regulatory element identifier. Actually, I must say these identifiers, they contain information about regulatory element position. So as you can see, actually, it has a organism ID, and then this is actually coordinates of CRE on the genome. So then it contains log likelihood score, motif, which interacts with this CRE, and then again it contains information about the closest transcript to the cis regulatory element. So, and yeah, here it contains transcript ID, gene name, gene ID, gene description, and yeah, this potentially might be interesting for some people. So it contains distance between transcript and regulatory element, and also association probability of regulatory element and the transcript. Finally, we have a motif significance table, and it's a bit different from what we have in ISMARA. It's extended in the sense that it includes, first of all, a column of motif names, then a column of significances, and actually this table is pre-sorted by significances, so the most important motifs you see on the top. But then, in addition, we actually have columns which correspond to the samples, and then for every sample and every motif, we have a Z value in this sample for this motif. So it basically contains significances, like shown here, and it also contains Z values for each samples. So also inside. All right. And finally, like for ISMARA, CREMA report contains all the information which you have online, like HTML pages, pictures. Actually, it also contains CRE information in different in GFF format, and binding sites predictions you can also find there. The only missing features with interactive features like gene search function, page which contains information about fraction of explained variants per promoter, and it doesn't provide averaging functionality. So like for ISMARA, for CREMA, data kept on the server minimum for 6 months. and yeah, user input data currently it's not removed right after the analysis finished, so it's also on the server. And now I think we can move to the last part of my presentation, to the averaging functionality of ISMARA and CREMA. So these functions are exactly the same between two servers, so I'm not going to present it separately. But before I start with averaging, are there any questions about data submission, upload, and so on? No, okay. Then let's go to the averaging. So what averaging does, it allows you to divide samples into the different subset, into the different groups, and then calculate average activity and corresponding average error bar for every group. Also calculate significances of motifs across the specified groups and identify regulators which have very little within a group, but quite a lot between the groups. So you can identify motifs which are consistently significant across different groups. And what kind of groups these are? So the simplest example replicates. So Eric has already shown example when we average replicates of the mouse liver data set. So you can do this to remove the noise between the replicates. Then another examples could be when you average by tissue type. This I am going to show later on. So in my case, I will show results for averaging between cancer and non-cancer tissues. And also, I think we also have it somewhere on example pages. Yeah, for one data set, it was possible to do averaging by age, because the subjects of the study were of different age, so you can actually also make such analysis. All right. So how averaging works? So here I try to do some illustration. So let's say originally I had 6 data points, and these are the pairs of replicates. And what happens after averaging? So it's basically going to average activity and corresponding error bar for this group, and I will get a single point instead of 2. And yeah, so I get activity and error bar recalculated, and now for this new data, for these new groups, I can again calculate the significance and find motifs which are important for the specified groups. So so how it works? So here's a little bit of math. So we assume that measured activities within the groups are coming from some unknown mean activity with unknown variance. So mean activity of the group and unknown variance in the group. And when probability to obtain the observed activity is given by this formula, and probability of our data given this mean activity and variance of the group is given like this. And by maximizing this probability over the variance, we can we can find the mean activity of the group and also the corresponding error bar. So exactly like shown on the pictures. So, and here's, yeah, again, example of averaging replicates for the, this is Illumina body map data set. So you see, so pairs of replicates are merged. And since the replicates are really good, so you see the original values are very close and error bars are relatively small, merging them actually produce results which have very small error bars, so it significantly improve the significance of the inferred inscription factors. And what I'm going to do now is to demonstrate how actually it works, exactly where person have to click, how to specify new sample names and so on. And for that, I'm going to use a data set which contains epithelial cells and also mesenchymal cells. So there are two replicates. So there is one sample of epithelial cells, two replicates, and three subpopulations of mesenchymal cells, two replicates. So I'm going to make a version. So let me switch the sharing window. Yes. All right. So this is the main page of this data set. And if you would like to perform a sample averaging, so all you need to do is just to click this button. And in this sample table, you will see some additional buttons and input fields. So first of all, you can enter your email address if you like to receive email notification once analysis is finished. Then you can also specify a project name. By default, this is original project name only with average prefix. And now we actually need to specify new subsets in the data set. So how to do this? So you need just to check a condition from drop-down menu. So you see, I'm going to specify it's going to be condition 1. This is also condition 1 because these are two replicates of the same condition. And you see when I click now with select box, I have now new option condition 2. So if I select it, I now have additional option condition 3. So you can always, specify more and more conditions if necessary. So I'm going to average replicate. So this is condition 2. This is condition 3. And this is condition 4. Also, for my convenience, I can specify the names of a new group. So in my case, let's call this is epithelial, and this is mesenchymal 1. So this is one subpopulation. This is mesenchymal 2, second, and this is third. And by the way, you see, when I type in one field, the name is changing also in all fields for the same condition. So in this case, condition 4, I have 2 times condition 4 chosen, and when I change in one field, it automatically changes in another one also. So it's going to be the third one. And I can give my address. And all I need to do just to click submit data for averaging. And now I'm again forwarded to the status page, like submitting data to ISMARA. And basically, if you haven't provided your email, you have to save the corresponding URL to check it later. Normally, running averaging jobs takes really a range of like 10-15 minutes, sometimes even shorter. So this is quite fast. Right. Let me switch back. I would like to talk also about another feature. And this is built-in batch correction. And meanwhile, actually, my averaging run is going to finish, I guess. Okay. So we also have one extra feature for averaging functionality, which is called batch effect correction. So the approach we use is extremely simple. And also we always say it should be used with caution, because potentially it might be dangerous to apply it to the data which do not require batch correction. So what it does, so in case, so let's say we had a data set, this is a kind of real story, which was done in 2 batches and 2 replicates per condition. And you see basically that there is, for example, in condition 2, this is extremely high variance between 2 replicates, as high as between basically conditions itself. But if you plot the same data by batches, so you can see that basically they're almost exactly the same. So they go up, go down, but there is a systematic shift between the batches. And in order to fix such situation, we apply a procedure which standardizes the data. So what it does, so before running averaging procedure, we actually take activities within a batch, and first of all, shift and rescale it by subtracting mean activity within a group and normalizing it by variance of activities within a group. And we also rescale error bars in this case, and we also rescale it by variance of activities within a group. And once we standardize data like this, so you see what is happening. So basically, it was rescaled and moved and shifted. And after this standardization, we apply the averaging procedure. And this I'm going again to show with the real page. Moment, how do I? Yes, I stop it here. Share my page. Sorry for delay. Yes. All right. Okay. Let's go back again to this epithelial mesenchymal transition data set. So in order to apply the batch effect correction, so first I again click on the perform sample averaging. I provide my email address. And you see there is a additional button, advanced option replicate batch correction. And in addition to the first column, which contains a condition selector, we now have a second column which contains batch corrector. And now I can select that, yeah, again, so it was condition 1, condition 2, condition 3. And again, I specify, so this is epithelial cells, this is mesenchymal 1, mesenchymal 2, mesenchymal 3. And now I can specify the batches. So yeah, they are actually indicated in the sample name. So you see A, B, A, B. So let's, so this is batch 1, this is batch 2. This is
You made an error, Mikhail.
no, it's not here. It's actually, actually it should be condition 3 here. Yeah, I overlooked it. And this is condition 4, and this is condition 2. This is condition 3. This is condition 2.
But isn't the second line a different batch than the first line?
No. So this is batch 1, this is batch 2.
Yes, okay.
Now these are batch 1, batch 1, batch 1. And these are batch 2, batch 2, batch 2. Yes. Okay. Now it should be correct. And yeah, all you'd have to do is just again click submit data for averaging. And yeah, after some time, you will obtain the results. So in this case, basically, there is quite good reproducibility between the replicates with low error bars, so we do not obtain much over averaging. But yeah, I think this small data set is really good for demonstrating. Okay, and the last thing to show, where is it? And the last thing to show is how to the results of averaging between different cell types. So this is example of how you can make a contrasts between different, for example, tissues or cell types in the data set. And we used for that a data set which contains 79 tissue and cell lines, healthy tissue and cell lines, and also 60 reference cancer cell lines. So altogether, it's more than 100 samples. And all these quite variable samples we have divided into 2 groups. So one contain only cancer sample, and another group contain only non-cancer sample. So this data set you can actually check from the examples page, from the example section of the main ISMARA page. And here I show a final results after the averaging. So you can see here I list the top motifs which were inferred after the averaging procedure. And so you see it indicates, so for example, this motif is going up in the cancers, this motif goes down, this up, down, up. And the interesting thing that, for example, these motifs which have, I think this is CAAT motif, yes, CAAT motif, I think. So it's well known that targets of these transcription factors are very highly expressed in the cancer. This one, which goes down, is very important for DNA repair and dysfunction of DNA repair mechanics, actually also one of the cancer indicators. MYC MECN and mixed transcription factors are very well known oncogenes, and you see they are very up in the cancers. This one was shown to be important transcription factor for cell proliferation and also extremely active in the cancer cell. So you see, even with very large and very heterogeneous data sets, with averaging procedure, you can obtain some very interesting insights on transcription factors, on regulators, which are driving changes between different groups of interest, which you can choose from the set of samples. Okay, thank you very much.